All right, so let's reconvene the meeting. And this afternoon we have on the agenda two speakers. They are not this is about basic research, not so clinical. We will start with Dr. Adriana Fontes. Let me find my notes. Adriana, as I mentioned before, she's from Portugal, but she works at the laboratory in Germany, in Neuherberg. And she's going to be talking about how the copper impairs the intestinal barrier integrity in Wilson's disease.
Everyone, I think right now you might be a bit tired, but hopefully you are well nourished from the lunch, so I think this will go well. So my name is Adriana Fontes, and I'm working since 2018 in Munich in the lab of Professor Hans Zischka. I guess many of you already met him, maybe. Actually, he's quite sad not to be here this year. He really likes Valencia and to come by. So we have been working a lot in Wilson, in copper accumulation, especially in the liver, and how it damages mitochondria. But today I will not talk about the liver, I will talk about the intestine. But before I start, I would like to challenge you to do a little questionnaire. So I'm sorry for the researchers, this questionnaire it's more for patients and caretakers and physicians. And it's anonymous, so you don't need to give any personal data, and it's very quick, it's not even 1 minute. So I hope it's working. You can access through the QR code, or you can go to the slido.com and insert that code. And I will give 1 minute, starting now. Hopefully it's working. It's working? Can someone? Okay. And in the end, we will be back to these questions and see the results, and maybe you can have 1 minute discussion. You can also answer during my talk, but I would prefer that you pay attention to the talk. So if you can, just answer during this 1 minute. 30 seconds. Ten seconds. It's the final countdown. Okay, so, but why study intestinal damage in Wilson disease? We know from the literature that around 40% of patients have abdominal pain at diagnosis. Also, this varies a bit from, for example, these are more European studies, for example, to the United States. But you can say between 20 to 40% of patients have intestinal complaints at diagnosis. Also, around 62% of patients develop gastropathy. Many times, ulcerative colitis is a comorbidity in Wilson disease patients, as well as celiac disease, especially in children with Wilson disease. And there are also some studies that show altered gut microbiota, so the bacteria that lives in our gut. What about patients? So here we have intestinal tissue from two patients. The first patient was diagnosed when he or she was around 8 years old. She was on the DPA treatment, but she complained a lot about abdominal pain. And then what this arrow is showing is the infiltration of lymphocytes, so of immune cells. So there was a high infiltration of these cells, so this patient had inflammation in the gut. In the second patient, this patient actually was first diagnosed with Crohn's disease, so inflammatory bowel disease, and only later on she got the diagnosis for Wilson, and she started on DPA treatment when she or he was around 15 years old. But we don't work directly with patients. We work with animal models, preclinical models. And here I show you two different models, one rat and one mice. And I saw exactly the same. So these animals, they have the ATP7B mutation. They have inflammation in their gut. So you can see this black arrow shows massive infiltration of immune cells in the rats and also in the mice. When I checked for different markers for inflammation, what I saw was that, especially when these animals are still healthy, when I mean healthy, I mean they don't have liver disease. So our animals, they start accumulating copper in the liver around 40 days old. Okay? Then when they are around 70 to 80 days old, they start to get disease, liver disease. And when they are around 120 days old, if they are not treated, they die. So if we don't treat them, they die, and the progression is quite comparable with patients. And what I actually see is that inflammation, it's higher when they are still healthy. So inflammation in the gut comes before liver disease. Then when we look at the mitochondria, so we know we have mitochondria failure in liver because copper mostly accumulates in the mitochondria because mitochondria needs copper for their function to produce energy. And when I look, so here is a nice mitochondria from animals that don't have Wilson, and here is the mitochondria of animals that actually have Wilson. So mitochondria is really damaged. And you see the same in the rats and in the mice. Okay? So here you have nice mitochondria with this cristae where energy is produced, and we have these dense areas, which means they're full of proteins. What happens in the Wilson disease rats in the intestine is that this mitochondria is damaged. Okay? And what we have in the lab, it's actually a cell line for the intestine, a human cell line that mimics this damage. So this cell line, we have also ATP7B knockouts, and what I do is I differentiate them to create a gut barrier, and then I look what's going on. And what I saw was actually the mitochondria of these cells, it's also pretty damaged. And one most interesting thing about the mitochondria, and you have here, on the right side, this is liver mitochondria, that we know it's highly affected in Wilson disease. And on the left side, you have mitochondria from the intestine. They are different, okay, already at basal level, so no disease. But when disease progresses, also the damage is different. So something is going on in the intestine, but it's different from the liver. And this, in the liver, you have this detachment of the membranes in the mitochondria, and the cristae, instead of being nicely together, form these kind of balls, which impairs energy production. That's why patients are also say they suffer from fatigue, so less energy. So we have damage in the intestine, but it's different. In the cells, it's the same. These are human cells, and what happens when I treat them with copper, so they are highly sensitive to copper, the ones with the knockout, the ATP7B knockouts, and they also, their mitochondria is less able to respire, is less able to consume oxygen and produce energy, as it happens in the liver, but here the damage is different. What about copper levels, right? We know that copper accumulates in the liver. What is happening in the intestine? It's also different. So, in the animals that are still healthy, meaning no liver disease, we actually see a slight decrease in copper content, which sounds strange at the beginning, but upon disease, so when the liver is damaged, we also have increased copper in the intestine. But, on contrary to the liver, this copper is not in the mitochondria, is in the cytosolic part of the cell. So basically, copper is floating around. It's not in the mitochondria, it's not in the nucleus, it's just in the cytosol. So we don't exactly know where this copper goes. Okay? But we know that this copper is enriched in vesicles. So it's not in the mitochondria, it's not in the nucleus, it's not in the Golgi, it's somewhere in vesicles. And we know that this is different in the intestine, in the liver, I'm sorry, because in the liver, it's mostly in the mitochondria. So we have intestinal damage, but we have a different way of managing copper. So between intestine and liver, there is a different way of managing copper accumulation. What about the barrier? What's going on? Here you have animals, so healthy animals, and animals with a mutation, but still with no liver disease. And what you see in this arrow is that there is an opening. So what you have here is two intestinal cells together, and in the wild types, they are nicely tied together, while in the animals with the ATP7B mutation, they have this opening. You can see it better here in the mice. So in both animal models, we have this kind of openings between the cells. And this you can nicely see, so this is an arrow that shows these openings. So these cells are not tied together. So these intestinal cells are not forming a good gut barrier. And this is just a measurement of this barrier, so to quantify. So it's nice to have pictures, because humans are visual animals, so we like to have pictures, but we need to quantify. And what we see here is that these knockouts have lower integrity. So they are less resistant to the passage of electric current. Is this what this graphic shows you after 21 days of differentiation? Okay. Then what we did, okay, we have an issue with these human cells from the intestine that have the knockout, the ATP7B knockouts. So we treat them with methanobactin to see, is it doing better? So we have been working on this compound for some years now, trying to push it also to clinical trials. And what we saw, it's actually got better. So the barrier integrity got better, so here we have more resistance. Mitochondrial function improved, so the mitochondria are better, are respiring better, are producing energy. And then this barrier, so here what it shows, these red lines, not sure if you can see, it's markers for proteins that are important to keep the barrier together. So this improved the methanobactin. In the CACO2 cells, there are human cell lines, but are, of course, an in vitro model. What about the animals? So we treat also the animals with methanobactin and look into the intestine. And what we saw was we treated diseased animals with methanobactin, and actually methanobactin was able to decrease copper content in the intestine, which DPA was not, okay? As you see here. And this decrease was mostly in that part of the cell that I told you about, that is the cytosol, where we suspect copper is in the intestine. And we also did a proteome analysis trying to understand, okay, what are exactly the changes in the intestine? What is happening? And what I want you to learn from this table is that what I found was inflammation in the non-treated animals. So again, one more hint, okay, we do have inflammation. And also we have the metabolism correlated with fat processing is also quite changing in the intestine. But the nice part was that when we treated methanobactin, actually there was a reduce in inflammation and an improvement in all this dysregulation of the lipid metabolism. So methanobactin is helping these animals overcome these issues in the intestine. To summarize so far, what we saw was, and actually the patients is in collaboration with colleagues in Poland, in Warsaw. They have a big group of pediatric patients, and they also saw inflammation in the intestine, the first pictures I showed you. And then we are working with 3 different models that we could see inflammation as well, together with copper mislocalization, also mitochondrial damage, as it happens in the liver with patients. And actually something interesting that we saw is that this transporter, this protein that brings copper in from the food, it's already very down in the intestine, even before the animals get sick. So the body of these animals, before liver disease, is already telling, hey, something is wrong. So it's kind of communicating to the intestine. You need to block copper uptake. So this transporter, that is CTR1, it's already down. And on the other hand, metallothioneins are already up in the intestine. So message is, even before liver damage, that I already said several times, the intestine is already on alert and already has inflammation. Okay? And when we treated with methanobactin, we saw so far some hints that it might help in this animal model, of course. But then you ask, okay, this is all very nice, but how can this help in the close future? So we have now a study approved to treat animals with zinc salts and to see what's happening in the intestine. Because we know that many patients get discontinued from zinc salts because they develop intestinal issues. What about if these patients already had some intestinal issues before? Maybe this is good to know what's happening in the intestine, so we don't prescribe zinc at all, and we try to find a better way. Okay? I think this is the most important message, and of course create some awareness towards the intestine in Wilson. And apart from trying to understand what these therapies are doing the intestine, we are also trying to understand a bit more about the mechanisms. Okay? Because then we can also understand maybe it's copper, is this inflammation going through mitochondria? Is the issue with the gut microbiota? Because since there is no copper going through the bile is altered. So what reaches the intestine is different. What's going on with digestion, with nutrient absorption, all of these things. What if it's copper per se, not through mitochondria and not through microbiota? What if it's copper that is interacting somehow with inflammatory markers, with pro-inflammatory cytokines? What if it's copper that's actually doing the work alone? This is what we are trying to know right now. And I would like to acknowledge all the people involved in this work. There are much more data, but this would be very exhausting. And maybe we could try to look now at the results of the questionnaire. Hopefully it works. Okay. So 55% of you are patients, 42 are caregivers, and 3 are medics or researchers. And then 34% of you never had, which is good. But some of you already had before diagnosis. 21% of you start to have an issue after treatment, and 50% of you. So remember in the beginning of my talk that I said around, okay, 40% globally, 16–70% in the US. So I think we are quite in this range here, so around 20% of you actually have intestinal issues. And however, there is no routine for exams, so maybe after today you'll talk to your physicians and ask, hey, can we check the intestine, what's going on? Maybe there's something I can help. Maybe there are some anti-inflammatory in the market, but this you need to talk with your physician, okay? Don't take this from me, I'm just a researcher. I work with rats and cells. Then, okay. So in here, when did you experience, no, not when, help. If, okay. So if you already have intestinal issues, what happened after? So there was no change, even with treatment, which means that also you didn't get worse, which is also good. Okay. Some of you, it improved with treatment, okay, which means that whatever copper is doing maybe got better with your treatment. But some of you, you are telling me that this got worse. And this could be due to treatment or due to some other things. The point is, the intestine is a very complicated organ, even more complicated than the liver. So there are many factors, okay? And this is the proof that environmental factors also play a role. Apart from medication, your day-to-day life, of course, nutrition, all of this will play a role in how your intestine, how your intestinal health goes throughout your life, okay? And this is what I wanted to know. Do you think it's relevant? That's why I wanted you to answer this questionnaire before my presentation, to know, still you think, okay, most of you think it's important to study, so I'm happy. To the 6% of you, I hope after my presentation you think, maybe we should give some credit to the intestine. Okay, thank you for your attention. Thank you for answering the questionnaire, and if you have any questions, I would be happy to answer.
Pasamos ahora.
All right, moving on. We are now going to have our last presentation of the day. We now have Dr. Pasquale Piccolo from the Teleton Institute of Genetics and Medicine, Pozzuoli, Italy, and he will be discussing gene therapy for Wilson's disease. Following this presentation, we'll open up a Q&A.